So thank you for joining us today. It's now my pleasure to introduce our speakers. Janalyn Frederick, Supervisor, Process Evaluation from Charles River Laboratories, and Artur Stanchek, Field Application Scientist, Process Chromatography from Bio-Rad Laboratories.
Thank you for the introduction. I'll be starting out our segment with a quick introduction to viral clearance and how we performed the Bio-Rad study. Viral clearance is the process of removing or inactivating viruses during manufacturing of biologics to ensure patient safety. We need to identify the potential to inactivate and remove both known viruses and unknown viruses. Biologics or biopharmaceuticals are medicinal products derived from human or animal sources such as recombinant products, blood products, vaccines, products derived from animal or human tissues and fluids, medical devices, therapeutical vectors, and gene therapies. In all of these cases, there's a potential risk for contamination. Viral clearance must be applied for all biologics. This slide shows an example general study design for a monoclonal antibody. This is performed at a downscaled model of the manufacturing process. A multitude of steps may occur during manufacturing. In this example, there are inactivation steps such as detergent treatment and low pH inactivation, and removal steps such as cation exchange chromatography, anion exchange chromatography, and virus or tenant filtration. However, for the study design for this Bio-Rad study, a mixed mode resin chromatography was used in place of the CEX and AEX steps. Mixed mode chromatography uses resins with multiple interactions. It can utilize methods such as hydrophobic interaction chromatography, anion exchange chromatography, size exclusion chromatography, and others. For this study with the Nuvia W'2A media, an AEX-HIC method was used. This will be discussed further by Artur in his segment of the presentation. Charles River Laboratories viral clearance is performed in two locations, Wayne, Pennsylvania and Cologne, Germany. We have reliably been performing viral clearance studies in Cologne since 1994 and Pennsylvania since 1998. Both sites have fully trained staff with multiple client laboratories for study use. Both sites are also able to perform HIV studies in either our BSL-2+ or our BSL-3 suites. The first stage of a study is to perform pretesting. Pretesting in general consists of a cytotoxicity assay and an interference assay. These tests are used to determine the non-cytotoxic, non-interfering dilutions to be used for the main study step. Cytotoxicity allows us to see the influence of the client-provided test sample on the cell line used for the assay. The goal is to find the non-cytotoxic dilution. We introduce the test sample neat and on a 96-well plate continue 1 to 3 dilutions across the plate to see if there is an impact to cell viability and phenotype. Once we find the non-cytotoxic dilution, we move on to interference. If you look at the bottom corner at the example plate, you can see the undiluted test item is shaded red for the first column, and as we go across, it gets closer to green. We would consider green the first non-cytotoxic dilution, and that would be the dilution used and moved on to the interference step. The second pretesting step is interference. Now that a non-cytotoxic dilution was determined, we can test to confirm if that dilution is also non-interfering. The interference assay tests the client test sample's ability to impact the virus. This could be increasing cytopathic effect or inactivating the virus. This is performed by taking the non-cytotoxic dilution of client test sample into media and filling a 96-well plate columns 2 through 12. Virus is added to the first column and diluted 1 to 3 each column across the plate, similarly to the cytotoxicity. The plate using test sample is compared to a positive control, which just uses media and virus. Less than 1 log difference between the positive control and the test sample plate is considered to be non-interfering and can be used in the main study steps. A traditional single virus spike approach has CRL spiking a single high titer virus into the test sample, which is the client starting material. We use a validated test system of that virus on its indicator cell line, for example, murine leukemia virus on PG4 cells. From the spiked sample, we take an aliquot to test by TC-ID50 assay on the indicator cell line. From there, the process is performed, whether it be chromatography, viral filtration, inactivation. At the end, the product fraction is sampled and assayed on the indicator cell line. The difference in log titer value of the load sample and the fraction sample is what we call the log reduction factor or the log reduction value. This is the number reported out to the client. In this example, the load had 8 logs and the fraction had 3 logs. The difference is an LRV of 5 logs. For this study with Bio-Rad, we used a co-spiking method, following the same procedure but with both MVM and MULV spiked into the same material at the same time. Because the viruses have different indicator cell lines, these viruses could be tested at the same time without impact to each other. In the example on the slide, the same process as a single spiking approach was used with a load sample being assayed by TC-ID50 at the start of the process, and the product fraction being assayed at the end. The samples would then go on both indicator cell lines, and the log reduction value would be calculated for both viruses. Now, Artur will discuss the chromatography method development for the study.
Thank you, Jenaline. Now, to start, let me introduce the mixed mode resins available in Bio-Rad's portfolio. The first and a very unique is ceramic hydroxyapatite resin. It has been in use for over 30 years and has a strong track record in commercial processes. The success of this mixed mode resin comes from its unique combination of calcium affinity and cation exchange modalities. There are different versions of CHT, which vary in pore size and bead size. It is best suited for polishing applications, and it's well known as a gold standard in removing aggregates. The second mixed mode resin I'd like to highlight is Nuvia C Prime. This resin combines cation exchange properties with hydrophobic interactions due to an aromatic ring present in its ligand structure. It is classified as a weak cation exchanger with a narrow pK range between pH 4.8 and 5.2. It has a 70 micrometer bead size and it's suitable for capture and intermediate purification steps. In our portfolio, we also have two hydrophobic anion exchangers, which share the same base beads but differ in their ligands. First of them, presented on the bottom left, it's a Nuvia A'4A. It has an aromatic ring on the end of the ligand and it's a strong anion exchanger due to the quaternary amine present. It remains positively charged across the entire pH range used in bioprocessing. Second, Nuvia W'2A, recently launched, it's a weak anion exchanger. Its ligand structure is identical to A'4A, except that it contains a secondary amine instead of quaternary amine. I will discuss how this affects ligand properties in the next slides. Successful purification doesn't depend solely on the ligand. It also requires the right bead chemistry and structure. It's important to note that all Nuvia resins share the same polymeric backbone, which is made of rigid polyacrylamide. This provides mechanical and chemical stability, allowing the resin to withstand concentrated acid, caustic, and denaturating agents. The mechanical stability ensures consistent process performance at high flow rates without compromising bed integrity. Additionally, the polymer is highly hydrophilic, which minimizes non-specific binding and allows for milder elution conditions, leading to higher yields. The open pore structure supports good dynamic binding capacity, even for large biologics such as viruses. This feature is particularly critical in virus clearance studies, as it allows model viruses such as xenotropic murine leukemia used in this study bind effectively in the resin pores even at high titers. Finally, all Nuvia resins have a narrow particle size distribution, eliminating the need for fine removal and providing good pressure flow characteristic. This slide presents the structure of our new hydrophobic weak anion exchange ligand, along with its pK ranges. One key property of the secondary amine in this ligand is that its charge state changes depending on the pH, making the ligand tunable and thus modulating selectivity. The secondary amine has a midpoint pKa of 7.4. This means that at pH 7.4, half of the ligands are charged and half are uncharged. In the uncharged state, the ligand primarily exhibit hydrophobic interactions. At pH 8.4, over 90% of the ligands are uncharged, making the hydrophobic interactions the primary contribution for molecule interactions. Conversely, at pH 6.4, over 90% of the ligands are positively charged. When comparing this weak anion exchange ligand to quaternary amine ligand, which remains permanently positively charged, we observed higher binding under the same conditions, likely due to reduced repulsion between some uncharged ligands and the product. Now that we understand how the new weak anion exchange ligand functions in Nuvia W'2A, we tested its ability to remove endogenous viruses. For this, we used a monoclonal antibody with an average pI of 9.2 expressed in CHO cell system. Before conducting the actual virus clearance study at Charles River's laboratory, we performed a process development study at our application lab in Germany. We tested three pH conditions based on the pK of the secondary amine ligand: pH 6.4, where over 90% of ligands are charged, pH 7.4, and pH 8.4, where over 90% of ligands are uncharged. Since hydrophobic anion exchange resins carry a positive charge, they are typically used in the flow-through mode, where impurities bind to stationary phase and the product flow through. We apply this method at pH 6.4 and 7.4, where the map is positively charged, ensuring sufficient repulsion between the ligand and the product. Chromatography runs were performed at moderate conductivity. At higher pH, repulsion decreases, leading to lower recovery but better impurity clearance. Impurities were removed using high salt washes and caustic sanitization. Developing a purification step at pH 8.4 was more challenging than at lower pH values. At this high pH, less than 10% of ligands are positively charged, and the map is near its isoelectric point, resulting in low repulsion. As shown in the top chromatogram, under low conductivity conditions, half of the product binds while the other half flow through. Since flow-through mode was ineffective at this pH, we switched to a bind and elute mode. To enhance hydrophobic interactions, we increased salt concentration using sodium chloride, although ammonium sulfate might be even better choice in some cases. By raising the sodium chloride concentration to 1 molar, we achieved strong binding. Elution was performed by lowering salt concentration to 20 millimolar sodium chloride and reducing the pH to 6.4, thereby decreasing hydrophobic interaction and increasing repulsion. This approach resulted in a yield approximately 90%. The optimized chromatography conditions were then transferred to Charles River laboratories for viral clearance studies. Runs at pH 6.4 and 7.4 were conducted in flow-through mode, while the run at pH 8.4 was performed in bind and elute mode under high salt conditions. All chromatography runs were performed using 5 ml prepacked columns at flow rate of 200 cm per hour, what resulted in a 3-minute residence time. For flow-through application, loading was approximately 85 mg MAP per milliliter of resin, whereas for bind and elute mode, loading was approximately 25 mg of monoclonal per milliliter of resin. This slide shows chromatogram from the actual virus clearance run. As pH increases, the charge of both the ligand and the mAb decreases, reducing repulsion and enhancing hydrophobic interactions. For virus clearance, we used MVM and MLV, which are negatively charged mother viruses, for rodent cell lines. Their charge is opposite to the ligand positive charge, enabling effective binding at all tested pH levels. Now, I will hand it over to Janelin, who will present the log reduction results for each conditions and virus tested.
Thank you, Artur. Now we can move on to the results segment. As you can see from the results table, all clearances were approximately 4 log and higher log reduction. If you'll notice, all results contain a greater than or equal to sign. What does this mean in viral clearance? It means that there was no residual infectivity detected in the N sample. This means that this is the minimum reduction for the N sample based on the detection limit of the assay. Now, this means that there could still be virus remaining, however, it is below detection levels. This is why it is stated that the results are greater than or equal to the number. No residual infectivity shows a robust removal or inactivation step. What is the significance of 4 log reduction value? The ICH Q5A revision 2 states that an effective viral clearance step generally gives reproducible reduction of virus load in the order of 4 log 10 or more, shown by at least 2 independent studies. Generally speaking, a reduction of 1 to 3 logs can also be counted towards total reduction for the process. Anything less than 1 log is not considered towards total log reduction values. Thank you. If you are interested in a viral clearance study with our Wayne site or have any additional questions, please reach out to pabvcquote@crl.com, myself at janelin.frederick@crl.com, or to Tarek Jaber, our Associate Director at tarek.jaber@crl.com. If you'd like to learn about any other Charles River services, please scan the QR code below.
Thank you for your time today. To learn more about Bio-Rad and chromatography resins, you can visit our resource site at biorad.com/library and request resin samples. Speak to our scientists and email us at process@biorad.com. We are happy to take questions now.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speakers now. So our first question is, in what circumstances would a co-spiking study be ideal?
An ideal time to use a co-spiking method would be when a client has a shortage in material, such as when producing a gene therapy, as sometimes those materials can be expensive and hard to produce. Another time would be if you're limited on equipment, such as chromatography columns, or if you're short on time and you don't have the luxury of running 2 viruses for 2 weeks. You can do all of it in one step.
And our next question is clearance of virus during chromatography necessary if viral inactivation was performed during protein A step?
I believe I can take this question, and the answer is yes, definitely. According to Q5A guidelines on viral safety, a specific limit must be met, and importantly, viral safety must be achieved using orthogonal methods. This is crucial. Simply repeating pH inactivation step is not accepted. So for products expressing mammalian cells, the downstream process needs to demonstrate approximately 13 to 15 log reductions of retroviruses. To achieve this, it's not just about applying low pH inactivation and virus removal filtration, but chromatography steps are also necessary. From low pH inactivation after protein A and virus removal filtration, 8 to 10 log reduction is realistic. The remaining virus removal must therefore be achieved through chromatography steps. That's why it's essential to use a resin that can support you in meeting your viral safety goals. I hope this answers your question.
Okay, and our next question is, are you working on any additional combinations of co-spiking other than the MULV-MVM combination?
Yes, we are currently working on a couple other combinations. We have a VSV baculovirus combination that is currently processing through our quality assurance team. We also have in the works an adeno S40 virus combination and an MULV adenovirus combination. These should be added to our offering soon, and in the Cologne, Germany site, they already have a couple combinations already available.
And what are the critical parameters that must be considered for setting up good viral clearance studies?
That's a very good question. In principle, critical parameters for virus clearance studies should be based on process development data and risk assessment. However, to some extent, they can be generalized. As we've seen, Nuvia W prime 2A can be used in both flow-through and bind and elute modes. In both cases, virus remains strongly bound, but critical parameters can vary. For flow-through mode, if we consider worst-case conditions, this would typically involve higher loading volumes, higher flow rates, wider collection criteria. If bind and elute mode is used, I would expect worst-case conditions to include lower binding, lower flow rates, and also wider collection criteria. pH and conductivity in most cases can also be considered as critical parameter. However, it's difficult in this case to define universal worst case conditions since they depend heavily on the specific molecule process conditions and if the chromatography is run in flow-through or bind and elute and which modality is predominantly used, HIC or anion exchange. But the good thing is that you can leverage on either of these modalities from Nuvia W prime 2A and get very effective virus removal.
Okay, well, it looks like we are running a bit short on time, so if we don't get to your questions, we'll be passing them along to our speakers who can follow up with you directly. So please go ahead and continue to type in any questions you have and we'll get those passed along. The last question for the webcast is it necessary to do preliminary testing before a viral clearance study?
I can take this one. Yes, we recommend doing pre-testing before your viral clearance main study. When using the TCID50 assay, it is important to determine what the non-cytotoxic, non-interfering dilution will be for the testing. TCID50 is a cell-based assay, and this pre-testing allows us to avoid repeats during main study.
Well, thank you, Janalyn and Artur.
Thank you for having us.
Thank you.